Good morning, my dear students. Welcome to my channel Patel Science Academy, and this is your Shahzad sir, and I'll welcome you in the lecture number 9 from the chapter Inheritance and Variation. Dear students, at the end of this lecture, you will be able to know about the crossovering, the Morgan experiment, and the other topic including the autosomal inheritance with their examples, widow's peak and PKU. Phenylketonuria. So dear students, let's start the lecture now, and let's start the topic is crossovering or recombination. So dear students, crossing over is actually a process that produces new combination. That's why it is called a recombination, because it is the process which produces a new combination by mixing from the both parents for the next generation. So crossing over is a process that produces new combination, that is recombination of the genes by interchanging and exchanging of corresponding segments between the non-sister chromatids of the homologous chromosomes. So dear students, it actually occurs during the Pachytene stage of prophase 1 of meiosis type of cell division, and the term crossing over, this was coined, means first time used by the scientist Morgan. So dear students, the homologous chromosomes, the one you receive from your mother and the partner that you receive from your father, they joins. So what is happening in the crossovering? The homologous chromosomes from the mother and the father, they join together to give you the recombination or new combinations. They exchange the genetic material, and the resulting chromosome passed to the offspring contains some of the paternal and some of the maternal chromosomes. So that's why it is the mixture of the both side, from the father side and from the mother sides. So yeah students, simply this is about crossing over, and the phenomenon of crossing over is universal, and it is necessary for the natural selection because it increases the chances of variation. So dear students, in short, crossing over means the joining of maternal and paternal side genes from the homologous chromosomes to produce the new combination, which is called recombination. So this is about crossing over. Here you can see the crossing over during the meiosis crossing over, that is also called recombination. The homologous pairs moves close together, and the chromatids may exchange the genetic material. As you can see here, the genes in this pair, the genes that have crossed over, you can see here the combination coming from these two chromosomes. So first cell division, what is happening during the first cell division? The homologous pair of the replicated chromosomes, they separate. So you can see here these pair is separated here, and in the sister chromatids now separating. So because of this separation from this pair, this sister chromatin, the 4 haploid daughter cell, they get it. So that's why it is the mixture. You can observe it from the different color, pink color and blue color. So you can see this pink color is coming here, and pink as well as blue color is also coming here in this one, in this two, and in this one too. So this is the new combination, or also known as a recombination, and it is only because of the crossing over that takes place in the homologous chromosomes gene to produce the new combinations. So now we'll see Morgan experiments. The Morgan, actually the full name of the Morgan is Thomas Hunt Morgan, and he was born an American. He was an American biologist, and he used a fruit fly that is called drosophila melanogaster in a genetic research, and established the chromosomal theory of the heredity. And he also discovered the principle of linkage, the sex linkage and the crossing over. So Dear students, Morgan's work played a key role in the field of genetics, and he was awarded by a Nobel Prize in 1933 in physiology and medicine. He was born in 1866 and died in 1945, and was awarded by the Nobel Prize in the field of physiology and medicine in 1933. So what was his experiment? So as you know, you can see here the drosophila fly. He used the drosophila fly for his experiment. So Morgan crossed two breeding, that is a red eye breeding, that is red eye female with the white eye male. So you can see from the upper side he crossed the red eye female with the white eyed male. So you can see here this is white eye male and this one is a red eye female. So as you can see here, the white eye is from the male side and red eye is from the female side dorsal drosophila fly. So what he got all red eyed filled when he crossed these two red eyed and white eyed male and female respectively. He got all red eyed flies in F1 generation. How it is possible? You can see here this is red eye, this is red eye, again these are also red eye. So he was surprised to get the result from this cross, because there were two parents, one with the red eyes and another with the white eyes, but in the F1 generation he got red eyed flies only. So what about the white eyed parents? What about the characters of the white eyed? So this Thomas Hunt Morgan, as I told you, got Nobel Prize in 1933, the winner of the chromosomal heredity in the Columbia University, and first to associate a specific gene with a specific chromosome, drosophila melanogaster, that is also called as fruit fly. So he was that brilliant man who discovered this thing. So Morgan and drosophila, actually this I told you. Let's see one more time about the introduction of the Thomas Hunt Morgan, first to associate a specific gene with the specific chromosome in early 20th century, and he used drosophila for his experiment, and he used the true breeding between the red-eyed drosophila and white-eyed drosophila. So female was red-eyed and male was with the white-eyed, and he got first in the F1 generation, first filial generation, all progeny was with the red-eyed. So good genetic subject. So dear students, as you know, in the 20th century, Thomas Hunt Morgan adapted the fruit flies Drosophila melanogaster, which is also called as fruit fly, as a model organism for the genetic research. And Morgan's first goal was to identify the different phenotypes, and he called the most common phenotype for each trait wild type, and then he then inferred that phenotypes that differ from the wild type resulted from a mutation or a change in the gene, and that was because of the crossing over. Individual with a trait attributable to mutation are known as mutant. So this was the Morgan experiment. Morgan, as you know, used Drosophila melanogaster, that is fruit fly, for his experiment because Drosophila can easily be cultured in the laboratory and its lifespan is short, about 2 weeks only. Moreover, it has the high rate of reproduction. That's why the choice of the Morgan experiment was fruit fly, that is dragon melanogaster. Morgan carried out several dihybrid cross experiments in the fruit fly to study the genes that are sex linked, and the crosses were similar to dihybrid crosses as carried out by the Mendel in the garden pea plant. For example, the Morgan in his group crossed yellow bodied, white eyed female to the wild type with the brown bodies, red eyed male, and intercross their F1 progeny. Then what he got? He observed that the two genes did not segregate independently of each other, and F2 ratio deviated very significantly from 9:3:3:1 ratio. So Morgan and his experiment knew that Morgan and their co-worker, they knew that the genes were located on the X chromosome and stated that when two genes in the dihybrid cross are situated on the same chromosomes, then the proportion of the parental combination is much higher than the non-parental type. So because of that, this occurs due to the physical association or the linkage of the two genes. He also found that when genes are found grouped on the same chromosome, some genes are strongly linked and they show very few recombination, that is 1.3% only. When genes are loosely linked, that is present far away from each other on the chromosome, they show more higher recombination, that is 37.2%. For example, the genes for yellow body and the white eye were strongly linked and showed only 1.3% recombination in cross 1. White body and miniature wings showed 37.2% recombination in cross 2. So cross 1 shows crossing over between the gene Y and W. Cross 2 shows crossing over between the gene white, that is W, and miniature wings, that is M. Here dominant wild type allele are represented with the plus sign. So this was that cross done by the Morgan and his group, and this shows the crossing over. When he crossed two F1 red-eyed flies, he got 3 red-eyed flies to every one white-eyed fly. Also the white-eyed flies were male. Why? Because of that crossing over. So experiments results for the Morgan was this you are watching here. Now we'll see autosomal inheritance. What is autosomal inheritance? The autosomal inheritance of a gene means that the gene is located on one of the autosomes, and this means that males and females are equally likely to inherit the genes. Dominant means that a single copy of the gene that cause a particular trait, such as the brown eyes instead of blue eyes. So dear students, autosomal inheritance, the human somatic, that is diploid cell, which is indicated by 2 small n, means 2n. So human somatic cell contains 23 pairs of chromosomes. They can be divided functionally as autosomes and sex chromosomes. A single pair of a chromosome is involved in the sex determination, and remaining 22 pairs are called autosomes. So dear students, in the diploid cell 23 pairs of chromosomes are there, out of which one pair is sex chromosomes, which decides or which determines the gender of the next generation, while 22 pairs are of autosomes. They are not sex chromosomes. So autosomes controls the variety of the traits other than the gender. So these traits are called autosomes linked trait, and the transmission of the body character other than the gender or sex linked trait from the parents to their offspring through the autosomes, it is called autosomal inheritance. So I hope autosomal inheritance has been understood by you. So what is autosomal inheritance? It is the inheritance of those 22 pairs out of that 23 pairs of chromosomes, uh, which was not responsible for the determination of the gender of that parent. So it decides the secondary sexual characteristics, that is the voice change, the development of the organ, the development of the pubic hair, etc. All that characteristics are called autosomal traits, and it is responsible for the inheritance of that autosomal character from the parents into their next generation, and that type of inheritance is called autosomal inheritance. So autosomal inheritance may be dominant, and it will be understood by this widow's peak and Huntington disease, etc. So we'll see it one by one. Widow's peak. What is widow's peak? Dear students, a prominent V-shaped hairline on the forehead of the person is described as a widow's peak and is determined by the autosomal dominant gene. So widow's peak occur in the homozygous dominant, that is indicated by the capital W this pair, and also heterozygous, that is capital W small w individuals. So widow's peak is a V-shaped point. You will see it in the next slide by the image. So it's a V-shaped point or hairline in the center of the forehead, and hair growth on the forehead is suppressed in a bilateral pair of the periorbital fields. Without a widow's peak, these fields joins in the middle of the forehead so as to give a hairline that runs straight across. So you can see here widow's peak in these two picture. This is V-shaped hairline on the forehead, and this is called widow's peak, and this is straight hairline. This is not called widow's peak. So this is that widow's peak you are observing here right now, and this is the autosomal dominant gene transmission. So widow's peak occur in the homozygous dominant and heterozygous dominant individuals. So it is indicated by W. Homozygous dominant gene is indicated by capital W, and heterozygous indication is capital W small w. So if person will have individual with the homozygous recessive condition, means the in the person, if small w means recessive gene pair is there, so that will have this straight line, means small w is there in the recessive condition, so this person will have this straight line, and this widow's peak will come if two capital W's are there, or means homozygous dominant condition capital W, and heterozygous condition capital W small w. So in this situation, if you are having this pair in your body, your hair will be widow's peak, either this pair or this pair small w capital W. If you are having this one too, so you will have this widow's peak in your body. If you don't have this type of pairs in the dominant condition, If you are having small w recessive condition, you will have straight line of your hair in your body. So it is present in both male and female because genes are present in both body. You can one more time see here widow's peak in this girl and straight hairline in this one too. So it can be easily observed. This is widow's peak. It means in this girl the pair will be either capital W or capital W small w. While this is not widow's peak, this is straight hairline, and in this body there will be small w. So because of this recessive condition, no widow's peak will be there, and because of this dominant condition, maybe in the homozygous condition or heterozygous condition, maybe in the same or different condition. So if the gene is in the dominant condition, this hairline will appear there as a V shape, and this is called widow's peak. You can also observe this widow's peak in this actress too. So this actress will have either capital W small w or capital W. Out of these two pair, any one pair will be there in this body. That's why this widow's peak is appearing here, but there will not be small w in her body. That's why this straight hair is not here. This widow's peak V-shaped hairline is there. So I hope you have understood about this widow's peak. Now we'll go for the autosomal recessive trait. Previously I have explained you about the autosomal dominant trait, that is the example was widow's peak, and I hope you have learned it well. Now we'll see the autosomal recessive trait. So it is an inborn metabolic disorder which is called PKU, phenylketonuria, and cystic fibrosis and sickle cell anemia. All these are the example of the autosomal recessive trait. What is phenylketonuria, which is also called as PKU? It is an inherited inability to metabolize the phenylalanine, which if untreated cause the brain and the nerve damage. So that's why this is called phenylketonuria or PKU, because this is an inherited inability to metabolize the phenylalanine. And dear student, it is an inborn metabolic disorder caused due to the recessive autosomal gene, and when recessive gene are present in the homozygous condition, this phenylalanine hydroxylase enzyme is not produced because of that recessive gene, and this enzyme is essential for the conversion of the amino acid phenylalanine into tyrosine. And due to the absence of these enzymes, which enzymes? Phenylalanine hydroxylase. Because of the absence of these enzymes, the phenylalanine is not converted into tyrosines. Hence the phenylalanine and its derivatives are accumulated in the blood and cerebrospinal fluid CSF. So the presence of the phenylalanine and its derivative in the blood and in the CSF, it indicates this disease, phenylketonuria, and it indicates that the pair of the gene is in the recessive condition, and it affects the development of the brain and cause the mental retardation. Excess phenylalanine is excreted in the urine, and hence this disease is called phenylketonuria, because it is found in the urine too, because excess amount of the phenylalanine is released from the body by means of urine. So that's why it is called phenylketonuria. The autosomal recessive trait appears in both the sexes with the equal frequency, and this trait tends to skip generation. So dear students, this is about the phenylketonuria. The symptoms of the phenylketonuria includes the learning disabilities, mental retardation, tremors or jerky movement of the limbs, skin rashes, that is called eczema, epilepsy, hyperactivity, behavioral issue. So these are the symptoms of PKU in the patient because of that uh, phenylalanine. Severe mental retardation, brain damage, seizures, mousy odor in the skin and the hair, lack of pigmentation, eczema, IQ of less than 50, aggressive behavior, negative mood, reduced dopamine level, hypomyelination, variability in the expressivity, more prevalent in the certain ethnic populations. For example, Turkish 1 out of 2600 is found. So these are the symptoms of PKU. Now we'll go for the next topic, that is called sex linked inheritance, in the next lecture, dear students. So thanks for watching my video. Thank you very much.